Alright, today, this is Alex Generous for KRB Radio, and I will be talking about adenoviral vectors. The first thing to note is that these are double-stranded DNA viruses that have a linear genome, and they've got, they're in the naked DNA family out of the various icosahedral forms. Alright, there's a bunch of different subgroups of adenovirus. We're not going to focus on all of them. The most common serotypes are in subgroup 3 and 2 and 5. And I think this whole presentation will revolve mostly around those two. Here, this is a phylogenetic tree, so this shows how related different species of a virus are to each other. And so this group C, which is what this subgroup 3 refers to, it's more commonly called subgroup 3 or C. And right here, you see add 2 and add 5. Again, these other serotypes have differences, but these are the most commonly studied, especially add 5. Add 5 was basically the first human one that was isolated, and so all research has focused largely on it. And the others are starting to get more, but they're not nearly caught up with the field of add 5 research. There's probably like 20-30 years of research to catch up on, maybe 40 now. Alright, so there are a couple of genes, E1 through E4, L1 through L5. This E1 gene is going to turn out to be very important. So there's a major late promoter, which means that late genes are under its control. It has a tripartite leader. It's not important to know, for our purposes, much about that, just to know that they exist and that they're in front of the L genes. And then these inverted terminal repeats, which I'll call ITRs, are at either end, represented by these triangles. Those are required for replication of the virus. So say, for instance, you wanted to make it unable to copy itself, you delete these ITRs out and only have the genome, but then you can produce infectious virus. This next part, this psi symbol, is the packaging symbol that tells the capsid proteins to put this genome inside of the virus and the viral proteins. So as a double-stranded DNA genome, this has a lot of coding capacity, so you have a lot of genes, like the L's and the E's. Notice E1A and E1B are two of the very first proteins. So this virion has an icosahedral structure, like we said. There's 240 hexon capsomeres that will have 6 hexons, and then 1 of 12 penton capsomeres are surrounded by 5 hexons. So this is the pentamere, or the penton that's got 5 hexons around it. This is a hexon with 6 hexons, the hexon capsomere. And there we go. The free pentons are shown here in this long strip is called a fiber. There's different ways to look at the structure from the outside. This is what these hexons look like in reality when this is taken under an electron microscope. For our purposes, what we care about is note that this is a very regular structure and that this genome is packed very tightly in here. So you're going to be limited in coding capacity to what this genome can already encode. So if you're going to put in something new, you have to take in out something old. So the major capsid proteins for ligands, the fiber protein, this will bind to the primary receptor, which is going to be the Coxsackie and adenovirus receptor, very cleverly named. So adenovirus uses an adenovirus receptor. And then the pentone base, this interacts with integrin basically as a secondary receptor and will cause entry into the cell. These little pieces are made up of those hexons we were talking about before. The icosahedral structure comes from the full range of these hexons into these larger triangle structures. So they bind, this pentone base binds to these alpha V integrins, and like I said, the CAR, Coxsackie, and adenovirus receptor is bound by this fiber protein. So this is just a brief go through of how adenovirus infection works. You'll have your adenovirus, it'll bind to its primary receptor, it'll undergo endocytosis. This is from the bound integrins clustering, and they mediate the viral endocytosis. So that's that pentone base is not shown here, but this is actually interacting with integrins on this surface. And that's this RGD motif, and that binds the alpha V integrins. Like I said, this binds CAR. So CAR binding, then the RGD motif on pentone bases binds onto the alpha V integrins, and then those integrins will cluster and mediate endocytosis. This will go into an endosome. That endosome will acidify, which will cause these fiber proteins to break off, and it will eventually cause release of the virion, which will then be transported to the nucleus, bind to the nucleopore complex, and release its DNA into the nucleopore during the process of disassembly. The important protein for this opening of this endosome during acidification is protein 6. I said fiber protein because the simplistic way to think about it is fiber protein gets ejected, that causes a breakdown that then releases protein 6. So now we're going to look at this as a vector, and it's known that these particular proteins, such as E1B, 55KDA, and E4-ORF6, will lead to cell death. Alright, so here's another overview. You bind to the cell, you enter the cell, enter the cytoplasm, get transported to the nucleus, enter the nucleus, make your new genomes, make proteins, then you release new virions, and during that process of release you will kill the cell. So the goal of vectors is to get some sort of transgene. Transgene just means a gene not from that particular source. So it could be as simple as trying to put in dystrophin to fix Duchenne's muscular dystrophy, or it could be as difficult as putting in a suicide gene for a cancer therapy. The last half of the viral cycle is incompatible gene therapy, with the exception of an oncolytic virotherapy, which if you've seen some of my other videos, you'll know I talk about fairly extensively. So this making the new genomes, new proteins, and then releasing the virions is essentially what you're cutting out. So this E1A, as I was talking about, is very important to almost every step of the virus. So if you're going to attenuate it or make it incapable of killing cells, you need to take it out, or if you want it not to have subsequent infection beyond the primary infection. So this was deleted out in the original experiments, and this is an example of a replication competent. You still have E1, and you still have packaging signals and ITRs, but you can replace that E1A and get replication defective. This virus up top will this is just wild type virus. This is, if you would go out and do a sick person, you would find this virus. This virus, however, you have removed E1A, so you won't be able to get replication of the virus, hence its replication effective. So only cells that originally get infected by this particular construct will have virus. It won't be able to spread beyond the initial cells. And CMV is a very common promoter, common strong promoter, and then GFP or luciferase are reporters. GFP is green fluorescent protein from bacteria, and then luciferase is also from bacteria and another way to image. Events in viral infection. So if you have an E1 deleted virus, it will successfully enter because you haven't altered the fiber or the penton base. It will still get in the acidic environment and break out of it because you have an affected proton or protein 6, and you'll still get entry in the nucleus because you haven't altered the capsid proteins. Your genome, however, will not cause viral proteins to be made because you have replaced their coding capacity. Alright, so viral vectors in general. Wild-type virus will infect and proliferate. Some of them are oncogenic. So that gives us the idea that we can co-opt that virus technology and remove pathogenic viral genes to make a gene delivery vector. If they're defective, how do you grow them up? And that is answered by two possibles. You can have a packaging or helper cell line, and this is a mammalian cell that supplies these viral proteins, but these viral proteins tend to be toxic and your titer is going to be low. Titer is the measure of the amount of infectious virus, so you're not producing enough of the virion. Or you can add a helper virus, which is another wild type or recombinant virus that you co-infect alongside. And this helper virus will supply the proteins. The problem is when you use a helper virus, you'll get some helper virus in your final sample, which usually means you have your gene therapy vector plus an actually pathogenic virus, which is not what you want. So to use this recombinant virus, you have to block packaging of helper cells or purify the recombinant from the helpers in some fashion, which usually you take out the packaging signal of the helper virus's genome. So we're going to talk a little bit about a packaging or helper cell line. This was done in 293T cells. E1 is deleted, but these 293Ts express E1. Meaning you'll still get E1's effect, even if it's an exogenous source providing it. You'll make genomes and proteins, then you'll release new virions and kill this cell. But in a normal cell, you'll see expression of the transgene product because it's under a CMV promoter and not part of the adenovirus life cycle. Even without E1, it still gets expressed. So this is thymidine kinase is a very common gene for cancer gene therapies. Basically, it causes apoptosis. So these are able to transduce non-dividing and dividing cells, which is important. If you're targeting cancer, you only want to target dividing cells, usually not non-dividing cells. So this is a mark against adenovirus as an oncolytic vector. It is episomal, meaning that it does not integrate with the host DNA, so you won't get it integrating into, say, some oncogenic gene like P53, but instead, or I guess knocking out P53 would be oncogenic, or you could integrate into MYC, which is a very common way to induce cancer. But this won't do that because it's not becoming part of your genome. The flip side is that it's not necessarily copied every time you have division between cells. So you'll only have the number of virus genomes in each individual cell as originally infected it. and then it will be diluted down by every subsequent division. But adenovirus has wonderful titers, 10 to the 12th, 10 to the 13th per mil, which is very high, so it's very useful. Way to build this, you have homologous recombination in rec bacteria. So you have your plasmids, and you have your cutting sites and your gene of interest over on this one. Then you can essentially cut this, combine the two, and then you'll get your recombination into the appropriate vector. And then in these 293A cells, after you've linearized and you've transferred, you'll eventually get these virus particles. Encapsulation signal and packaging signal mean the same thing. Now you can see these ITRs. L probably refers to some modification they've made. You'll see the Ad5 DNA is here, but your gene of interest and its unique promoter are here, so you can get cell-specific activation. So the first generation of ad vectors were deleted for E1, so you could get up to 2 kilobase pairs of insertion of your transgene, meaning this is good for very small genes, but larger genes or gene cassettes can't be done by adenovirus, or at least not adenovirus in its natural state with a gene we put inside of it. I realized I didn't explain this. E3 can be knocked out, and it's shown here, without hugely detrimental effects to the virus. So if you're going to knock out something in adenovirus to be able to put a transgene in, E3 is a pretty good choice. So that's entirely a space-making move. It's not because E3 is super necessary. That allows up to 7 to 8 kilobase pairs, which is much better than just 2. But these first-generation adenoviruses are totally immunogenic. Just think about you got a cold probably fairly frequently throughout your life, and now you're using that same exact virus. Your immune system sees the same thing, even though you've changed its genome. So these first-generation ads and AB for propionic acidemia. You can see this is a higher treatment, and it appears to be working better. But you're getting a lower MECs. Adenovirus vectors, first generation, we talked about. Important to note, those are made in 293 cells, as opposed to a different type of culture cell, which may produce different results. Most first generation adenoviruses are deleted to make space. We just talked about that. And the second generation of vectors is deleted or temperature sensitive for E2 and E4. E2 and E4 need to be supplied by helper cells or viruses or plasmids, and they're engineered primarily to remove viral antigens to avoid this immune response. So basically, until these get to the right temperature, they activate, but when it gets too warm or something, they will go away. And apparently they help stop the immune response, probably by not presenting as antigen as commonly. There's also some small increase in insert capacity, so the most we're working with is 7 to 8 kilobase per machine. Upwards limit is probably 10 based on these notes. And then there's helper-dependent or gutless adenovectors, which have no viral genome, but rely on other viral genomes to get them packaged. So we have our replication competent, defective, and then defective helper-dependent. So you'll get human stuffer DNA, which might just be human DNA that doesn't do anything for protein, so that you don't accidentally come up with new epitopes to find those genes. Wild type Ad5 is E1, packaging signal ITRs. Then this gutless vector has no E1. It's also full of human DNA, so it's the dystrophin gene plus human stuffer. And these are both 36 kb, and then genes per cDNA are up to 35 kb. Gutless or helper-dependent vectors. All viral protein coding sequences are deleted from vectors. Viral protein supplied in trans from helper adenovirus. So this adenovirus is providing your gene therapy vector with the viral proteins it needs, but your gene therapy vector won't be able to provide any of the proteins it needs to itself. Hence, you can have a virus like this one that are gutless and don't have the viral proteins, just the packaging signal and the replication signals from the ITRs, which are necessary to propagate themselves. So you can remove viral antigens to reduce the immune response, and then, as I said, the removal leads to up to 35 kb of space. So if they're defective, the other way to grow them is with helper virus, which we got into a little bit. So you have your gene with the packaging signal and the ITRs on either end. 293 cells will be where some of these are grown, and that'll supply E1. So wild type helper E1 deleted can also be the helper, depending on if you want it to replicate or not. And it supplies everything except E1. E1 is probably added out in a different plasmid. The problem with these is that you will get some amount of normal adenovirus out of it, hence the green picture 2 adenovirus. That is not a gutless vector, that is a gutted virus, a non-gutted virus that still has all of its bad effects. The Cre-LoxP gutless adenoviral vectors. This is where you put LoxP sites on either side of something, and these Cre cells reply E1 and Cre recombinase. That means the Cre will excise this packaging signal out, leaving only one LoxP site, meaning this genome won't be packaged. Helper viruses may supply everything except T1. There is a preferential packing of the vector to less than 1% of the normal virus, but you don't want to have any normal virus. It's hard to get FDA approval for that. Helper-dependent adenovirus can give long-term expression after one delivery, so you can get just essentially one injection of this, and it should help. Transgene expression up to 7 years. Non-human primates following hepatic transduction with helper-dependent adenoviral vectors. So you can get this expressing for some amount of time, though it's not the best. Retrovirus and lentivirus are better for long-term expression. So for adenoviruses, the first generation were deleted for E1, Most first generation of adenovirus did that to make space. Let's see. The problems are that these are very immunogenic, the first generation adenoviruses, and then the second generation is the plan between E2 and E4 based on the cell or virus or plasmid or ambient heat, engineered primarily to, these were all engineered primarily to avoid viral antigens from coming into contact with the immune response. There's also some small increase in insert capacity based on removing those. And then gutless means everything's removed. Cell binding, we talked about CAR on the protein. Back to issues about vector tropism. Let's see. So this is just showing off CAR and the fiber protein. Add 5, and then a couple of the others binding CAR and alpha V integrins. So a lot of CD generally refers to some type of immune function, and then integrins and desmoglein are part of I forget, tight junctions or adherence junctions, one of the two. Desmogleins are neither. They're in desmosomes, but integrins are one of the two camps. But they're basically cell adhesion molecules, so how cells stick together without separating. And this virus appears to target those. So what happens in vivo? A single injection of replicating adjuvant luciferase in mice. You get a nice little picture. Why does adenovirus go to hepatocytes? Wow. So these clotting factors is protein C, factor 7, factor 9, and factor 10. So basically the adenovirus gets stuck in the liver by the filtering mechanisms, especially because these clotting factors can get pasted onto it. Yeah, so this slide is just showing how C8, 9, and 10, or C7, 9, and 10 can cause this liver targeting. Okay. This is just a graphical representation of the virus. Yeah, and this is after single injection. And see, without warfarin, but plus predosing, causes quite a bit of replication. Or not necessarily replication, just infection. So the largest problem is that AD5 has the largest pre-existing immunity pool. I think only probably influenza, or not influenza, measles is probably the only thing that gives it a run for its money. Serotype switching with HD adenoviruses. So there's 24 kilobase pairs of human stuffer. After that, there's a CMV promoter with the HIV on protein. So add 1 can be E1 deleted helper, add 2 can have the same thing. So you can switch between these serotypes, and it's very cleverly called serotype switching. So this is testing to pre-immunize macaques with AD5, and then serotype switch to an HIV vaccine. So the first thing to note is that 612 is very high, and then days after this you can see that the controls are doing better than either the exercise group or the non-exercise group. So I'm mistaken. The exercise group has nothing to do with this. I'm thinking of a different project. I apologize. So after this challenge, you'll get a higher viral load in the controls, which you would expect because you would expect a lower viral load after your vaccine here takes hold. So these are antibodies that you're finding, so you're getting an immune response, and this 612 means that's the least number of viruses, meaning it's getting cleared by the immune response, as you can tell by looking at the full control, fully immunocompetent host, and it is doing poorly at clearing this virus. So, why would you use adenovirus? Because it gives very robust expression, it's very high titers, it's episomal integration as opposed to actually integrating into the genome. The only downside is it gets cleared by the liver, and if you have enough viremia, that's the amount of virus in the blood, you can kill an animal, and I'm counting humans as animals in that. Again, remember that this can give long-term expression after one delivery, though you do see a steady drop-off, and this is transgene specific, so some genes don't work very well. And this large gene capacity, like 36 kb, and that's as compared to growth hormone only has 1400 base pairs, and dystrophin, one of the larger genes in the human genome, has 2.4 million. But its mRNA copy of 14,000 fits within this 36,000 restriction, so you can use this system to make dystrophin for those afflicted by DMD, Duchenne's muscular dystrophy, which I believe I mentioned earlier, but I'm reiterating now because it's been a bit. So adenovirus has immune and toxicity problems, and so this is a very famous case of Jesse Gelsinger, and he was an ex-SCID patient who basically died because his own immune system overreacted. And that's the end of the presentation. Special thanks to Mike Berry for preparing this lecture. If you'd want to contact me, Alex Generous, you can reach me at krebradio@gmail.com. Tune in to KRB Radio, which you can find on iTunes. Feel free to give us a rating. We hope it's 5 stars, but leave us comments, your views, what have you, if you disagree, and we'll try and make the show better. Thank you very much.